Last time we have discussed introduction to metabolism. What is metabolism? Metabolism is the entire spectrum of all chemical reaction inside the body. Metabolism is divided into three pathways: the catabolic pathway, anabolic pathway, amphibolic pathway. What is catabolic pathway? In which the large and complex molecules converts into simpler molecules, or the energy rich molecules converts into energy poor molecules. What is anabolism? And in anabolism, the small molecules, the entities or the building blocks combine with each other, react with each other, and form macromolecules or complex molecules. Mean from energy poor molecules, energy rich molecules are formed. Right? And what is amphibolism? Amphibolism is the metabolic pathway in which the catabolic and anabolic reactions occur simultaneously and same time, right? Like Krebs cycle. What is the example of catabolic reaction? Glycolysis, glycogenolysis are the examples of catabolic pathways. What is the example of anabolic pathway? Gluconeogenesis, glycogenesis, the formation of glycogen, formation of glucose. These are anabolic pathway. What is the best example of Krebs amphibolism? Krebs cycle, because in Krebs cycle both the anabolic and catabolic pathways occurs in same time. Right? So these are the examples, or these are the parts of metabolism. Now today we will go for the metabolism of carbohydrates, which is the first topic in your curriculum. In this topic we will discuss the carbohydrates metabolism, mean glucose metabolism, as well as sidewise we will discuss the fructose, mannose, maltose metabolism, right? And how these for metabolism of carbohydrates, the glucose must leave the plasma and will enter into cell or tissues, and in tissues the glucose will undergo metabolism. So we will also discuss the pathways or the proteins or the transporters which are responsible for taking the glucose from the blood into the cell, from the cell into the blood, also from the blood to the interstitial fluids. Because for example, I have जी हसीना, we are discussing carbohydrate metabolism. Carbohydrates metabolism, right? For carbohydrate metabolism, first of all we will discuss the intake of carbohydrates into the body. From where carbohydrates occurs? Carbohydrate is coming from reaction in green trees. We have discussed this yesterday, the photosynthetic reaction. Photosynthetic reaction. In photosynthesis reaction, photosynthetic reaction. What is the location of this reaction? Green plants or trees. Right? In this reaction, carbon dioxide react with water in the presence of sunlight and form glucose C6H12O6, which is called glucose. Inside the body, the main source of energy is glucose, but grossly the main source of energy is sun. And from this reaction, oxygen is released. Right? So glucose is coming from photosynthetic reaction. Now, for example, this glucose will store in fruits. It will store in fruits. Also it stores in vegetables. Also it stores in seeds. and human beings are feeding on fruits, vegetables and seeds, right? So for example, in fruits we have, for example, apple. We are eating apple, and this apple stores glucose in the form of, plant store carbohydrate in the form of starch, and animals stores the glucose in the form of glycogen, right? So here for example we are eating that apple which store glucose, right? And we digest by our digestive system, and this apple is again converted into glucose inside the body or inside the stomach. So we have abundantly glucose is now present, which came from this apple or from sunlight or from trees, right? Now from here this glucose will absorb into portal vein, and portal vein will take these glucose into liver. Why in liver? Liver is the main part in the body or the main organ in the body which is responsible for metabolism, any type metabolism. And I have discussed you that what is metabolism. The conversion of active things into non-active, or non-active into active, or toxic into non-toxic, this process is called metabolism. It is also known as biotransformation. The metabolic processes are also called biotransformation, because in this reaction the biomolecules are converted from one type to another type, mean from toxic into non-toxic. This is one type, this is one type, right? So this is also called biotransformation. The enzymes which are present in the liver, which are mainly responsible for metabolisms, are the family which is called cytochrome P450. The enzymes which are responsible for metabolism, mainly drugs metabolism, is cytochrome P450. This is enzyme family, right? And this liver is composed of which type of cell? The cells of livers are called hepatocytes. So for example these are hepatocytes, right? And this green is portal vein. What is the role of portal vein? It contains partial oxygenated blood, and it gives the blood from the GIT into liver, right? So what is the main role of the portal system or the portal vein? To take the blood as well as the nutrients from the GI tract into liver. It will take all the nutrients. For example, we are eating proteins. The proteins will convert into amino acid, and these amino acid will take by this portal vein and will transfer it into liver, right? But in this topic we are discussing carbohydrate metabolism, so we are only focus on glucose, right? From here, what is the normal sugar level of body or blood? Fasting blood sugar. It is from 70 to 110 mg per dl. Remember this or keep it in your mind. Normal blood sugar in fasting state, it will be from 70 to 110 mg per dl, right? So this liver is responsible for maintaining this sugar level constant in blood. How? Because we are taking glucose abundantly and more quantity, and if all the glucose will go into the circulation, here this portal vein divides, and here it form hepatic vein, and hepatic vein will goes into 4-chamber organ in our body which is called heart, and after heart the circulation is called systemic circulation, because from heart this blood will go to all our body parts, to brain, eyes, all body parts, right? And I have told you that the enzymes which are responsible for glycolysis are present in each cell of the body. In each cell of the body there are the enzymes which are responsible for glycolysis. So this glucose will go to all tissues of our body, but there are some specific regions or specific organs in our body which are wholly solely dependent upon glucose, and the organs are brain and retina, right? So from here this glucose level will be high in portal circulation, because it takes all the glucose from the GI system. But here when the glucose is reached to liver, the hepatocytes take glucose, and fortunately insulin are not needed for this transport. Insulins are not needed for the transport of glucose in the liver, right? So the liver takes the extra glucose from the blood, and only 70 to and only 110 to 170 mg of per dl remains in the blood, because our random blood sugar level is from 110 into 170, right? Here this is in fasting state. Why we are focused on fasting state? Because now we are only eating and the body is in fasting state, but when we eat the body is in random condition, or it start metabolism, right? So this liver takes the extra glucose from the blood and prevent hyperglycemia, because when sugar level is more than, for example, sugar level is more than 170 milligram per dl, this condition is called hyperglycemia. And here in fasting condition, when the blood sugar level is greater than 110 mg per dl, it will called hyperglycemia, right? So here this liver is responsible for preventing hyperglycemia, but this liver is also responsible for preventing hypoglycemia. How? When, for example, we have liver here, this that same liver, and here we have hepatocytes, and from here it stores glucose in the form of glycogen. For example, in systemic circulation, the sugar level become less than 110 mg per dl, our sugar level is less than 70 mg per dl. This is in random condition, this is in fasting condition. This condition is called hypoglycemia, and in this condition this hepatocyte will convert the glycogen into glucose and will give this glucose into blood, so the sugar level will be increases here. So the liver is called the sugar monitor of our body. It is the sugar monitor of our body, which monitoring the sugar level in the systemic circulation and converts or starts glycogenolysis or glycogenesis. When the body is in hyperglycemic condition, the liver will start glycogenesis, the formation of glycogen from glucose, and when the body is in hypoglycemic state, the liver will start glycogenolysis and will convert again the glycogen into glucose and will increase the body sugar level. So we say that we can say that liver is responsible for preventing hyperglycemia as well as hypoglycemia. Right? From here, this glucose will goes into, for example, this is systemic circulation. From here, I have, for example, this is the heart. This is heart, and from heart, aorta is arising from left ventricle. This is aorta, for example, and this aorta supplies the blood into all the body parts. For example, this is the blood, and in this blood now the glucose is present. In this blood the glucose is present because we have eaten glucose. So all the glucose is present in after fasting from liver. The only needed amount of glucose is present in blood plasma. Right? Here we have body tissues. For example, this is the tissue from brain. For example, this is the tissue from muscle. For example, this is the tissue from eyes, etc. But here one point is very important. What is the concentration of glucose in blood? What is the concentration of glucose in extracellular spaces? Because all the things, anyway, this is the drug, hormone, enzyme, all the things. First it will go to interstitial spaces, and from interstitial spaces it will go to cell. So these are the three compartments of the body. One is blood plasma, one is extracellular fluids, one is intracellular fluids. Right? So the blood glucose level will be different, and for example, the process diffusion. What is diffusion? The movement of molecules from high concentration into lower concentration. So for example, here the concentration is low and here the concentration is high. The glucose will go to inside intracellular fluid. For example, here the glucose concentration is high and here the concentration is low. The glucose will go from shifted from high concentration into lower concentration. But here this is not simple diffusion. It is the type of facilitated diffusion. It is a type of facilitated diffusion because some transporters or some proteins are responsible for taking this glucose into different compartments. But fortunately here in extracellular fluids and in blood plasma, the glucose level is same. Glucose level is same because if you remember the body fluids which we have discussed in your first year, body fluids, I have told you that extracellular fluids is divided into two parts. Extracellular fluids are divided into two parts. For example, this is blood and this is cell. This area is called intracellular fluids, and here you all know that here are the RBCs are present. RBCs. So outside these RBCs, this area is extracellular fluids, and outside these cells, this area is extracellular fluids. These two compartments are the two components of extracellular fluids. This is called plasma. In blood, the extracellular fluids are called plasma. In all other tissues, this extracellular fluids is called interstitial fluids, right? And this extracellular fluid and this extracellular fluid, it freely move to each other. For example, from here it can move to interstitial fluids, and interstitial fluids independently can go into plasma. Right? So for example, here in the blood plasma, glucose is present and these two are same compartments. So here and here the glucose amount will be same. If someone asks you in paper or in an interview or in viva, what is the concentration difference between the interstitial fluids and blood plasma, it will be same, because these both are extracellular fluids and can freely move into each other, right? But here in intracellular fluids, this concentration never be same, right? Because this glucose needs a well defined transport system by which this glucose can enter into the cell, right? And this is the way, this is the point where we have discussed, or we will discuss, the glucose transporters, the entry of glucose from the extracellular fluids or from the blood into the cell. Because in our body there are different types of cells. We have muscle cells, we have bone cells, we have RBCs, we have WBCs, we have neurons, we have retina cells. These all the cells are different from each other, and all the cell have different mechanism for glucose intake, right? So from here, for example, this is the extracellular fluid. From extracellular fluid, for example, this is the glucose. This glucose cannot enter freely into the cell. According to your book, the concentration difference here is, inside the cell the glucose concentration will be less than 100, right? So it will need a very well defined transport system. Now we will go for the transport system of glucose, by which the cells takes the glucose from the extracellular fluids into the cell, right? The topic is the entry of glucose into cell. Entry of glucose. There are two types of entry system to the cell. The first one is glucose independent pathways, and the second one is glucose dependent pathway, or facilitated diffusion and sodium glucose co-transporter system, right? So here, for example, entry system of glucose to the cell. First we will discuss insulin independent system, or transport. Insulin independent transport. Second is insulin dependent transport. Why we are discussing these topics? Why we are not directly discuss glycolysis? Why? Because there are, if the cell doesn't have glucose, can the cell undergoes glycolysis? No. So first we will discuss how the glucose enters into the cell, and after that we will discuss glycolysis, Krebs cycle, electron transport chain. That processes are very simple, right? Insulin dependent. Next transport system is facilitated, or we can say that these two transports are called facilitated transport. And another transport is sodium glucose co-transport, right? Now up to now there are, what is facilitated diffusion? Anyone? What is facilitated transport or diffusion? Anyone? Carrier pake, which need carrier. What is simple diffusion? From which doesn't have any carrier. What is osmosis? Semi-permeable membrane, which pass from the movement of molecules from high concentration into lower concentration through a semi-permeable membrane is called osmosis. The movement of molecules from high concentration into lower concentration area by using a protein or other carrier, this is called facilitated diffusion. The movement of molecules from high concentration into lower concentration is called simple diffusion. What is active diffusion? The movement of molecules from lower concentration into high concentration with the use of energy is called active transport. Right? So here we will discuss these transport, and this insulin dependent and insulin independent transport system is facilitated diffusion or facilitated transport. Right? And in kidneys and nephrons we have sodium glucose co-transporters, and there goes sodium glucose co-transport. Right? Up to now we have 6 proteins are discovered which are used for transport of glucose from the blood into the cell. The 6 transports molecules are, they are called Glut receptors or glucose transporters. Right? Glut 1, second is Glut 2, third is Glut 3, Glut 4, Glut 5 and Glut 7. Glut 7. So 1, 2, 3, 4, 5, 6. And different Glut receptors are present in different areas. Different cells need different Glut receptors. Right? For example, Glut 1 and 3. Remember with yourself, Glut 1 and 3. These two are present abundantly in body. And all over the body, the most Glut receptor present is Glut 1 and Glut 3. Specifically, these Glut receptors are present in the areas which needs more glucose, which are fully dependent upon glucose. Like for example, this Glut 1 and 3 are present in brain. Next, they are present in retina. They are present in placenta. Right? These are the areas where Glut 1 and 3. Brain mean they are present on neurons, because neurons are fully dependent upon glucose and oxygen. Right? The Glut 4 are present in, we will discuss Glut 2. Right? But Glut 4 are present in, these are the most important Glut receptor which are present in the area which can store glucose for future use. For example, muscle cells present in muscles cells. Also they are present in fats, mean adipose tissues. Adipose tissues. Right? And the only Glut receptor which is insulin dependent is Glut 4. Highlighted. This is insulin dependent. The only Glut receptor which is insulin receptor is Glut 4. All the other Glut 1, Glut 2, Glut 3, Glut 5, Glut 7, they are insulin independent. So these 1 and 3 are insulin independent. Insulin independent. These 5 and 7 are also insulin independent. Right? What is where GLUT2 well present? GLUT2 well present in the areas which can take the glucose freely from extracellular fluids into the cell and can give the glucose back into the interstitial fluids, right? Which can freely move between two-way pathway, which needs two-way transport. For example, hepatocytes. For example, I am taking here one hepatocyte. This is hepatocyte from here, and this is the blood supply, the portal blood supply, right? It can take glucose when it is in extra amount. It can take glucose easily, which the glucose is present in extra amount, and when the glucose level is decreases in the blood, it can give glucose back to blood. So it's mean, for example, here is the Glut transport system. Glut transport system, it follows the two-way pathway, right? This two is showing two-way, right? This is two-way pathway and present in mainly in liver. For example, here this is also insulin independent. Glucose, GLUT2 are also insulin independent. GLUT2 are present in liver, also it is present in kidney. One another very important location for the GLUT2 receptor is the beta cells of pancreas. Generally, we say that GLUT2 receptors are present in pancreas, but there are specific cells which are responsible for insulin production are having GLUT2 receptors. So the third location of the GLUT2 receptor is beta cells of pancreas, because this beta cell is responsible for insulin production and it regularly sucking the glucose level in body. So if the glucose level increases, it will release insulin and will lower down the glucose level, and if the glucose level is decreased in the body, the alpha cell will release glucagon and will increase the glucose level in body or increase the glycogen breakdown, right? So these GLUT2 receptors are present in liver, kidney and beta cells of pancreas. Now this is also insulin, these are insulin independent, these are also insulin independent, right? GLUT5, the next is GLUT5 receptors or GLUT5 transport. GLUT5 are not glucose transporters. These are not glucose transporters, these are fructose transporters, right? GLUT5 are fructose transporter, like brain, which like the glucose mostly and eat glucose. Some of our cells only eat fructose. What are these cells? First of all, testes. It's feeding on fructose, and sperms move with the help of energy which is released from fructose. So sperm cells also need fructose. So on sperms and testis cells there are GLUT5 transport, and these transports are also insulin independent. Insulin independent, right? The last one is glucose 7 transporter or GLUT7 transport system. This system is present in, or these transporters are present in endoplasmic reticulum, because endoplasmic reticulum is responsible for the metabolism of glucose 6-phosphate or other 6-phosphate, because phosphatase enzymes are there, right? So GLUT7 receptor well present on endoplasmic reticulum, and these are insulin independent. The only and only which is very important, which is insulin dependent, is GLUT4, and GLUT4 are present on muscle cells and fat cells, right? What will be the structure of these transporters? The insulin depend, what is the difference between the structure of the insulin dependent transporters and insulin independent transporter, right? So we are taking the cell here. For example, I am taking one cell from brain or from retina, from placenta, but generally, for example, this is a cell, and here I have blood. This is blood, and this blood contain a lot of glucose. Also, the glucose is present in interstitial fluids. Here, glucose is needed. Glucose is needed. So, the transporter present here is like this. It is like this, and it will attract the glucose towards itself. So for example, glucose came and stored here. When the glucose is needed inside, this protein, this facilitated intrinsic proteins, undergo some conformational changes. And what will be the conformational change? It will become like this, and will enter the glucose from outside into inside, outer side into inside, right? This is the transport system of insulin independent. This is insulin independent transport, right? For example, this is the transport system. This is like this, and glucose is, for example, these are glucose, and glucose are stored here outside. When the cell needs the glucose, it will do some conformational change like this, and it will give this glucose inside. This is insulin independent transport. And insulin independent transport systems are GLUT 1, GLUT 2, GLUT 3, GLUT 5, and GLUT 7. These are insulin independent. The insulin dependent, for example, what this glucose will goes inside. For example, this is 6 carbon compound. It will undergo glycolysis and will make 2,3 carbon compound, which are called pyruvates. Right here, for example, this is another cell. I am taking the cell from muscle. This is the cell from muscle, myocyte. Right here, for example, I have the blood. This is the blood, and this blood have a lot of glucose, and when the glucose level is increases in the blood, insulin will be released by pancreas. So for example, pancreas forms insulin, and insulin is also present in blood. Right here, the GLUT4 transporter, which are insulin dependent, they are not present like on surface of the cell. These are present inside. For example, naturally there are some vesicles inside the muscle cells and the fat cells, and these vesicles contain some transporters which are called GLUT4 transporters, right? And when these vesicles are present inside the cell, it cannot work. It cannot take these glucose from the muscles from the blood into the muscles. So what will happen? There are insulin receptors. Do you remember one pass receptor? I have taught you one pass receptor, insulin receptors, 7 pass receptor, 12 pass receptor. Here are the one pass receptor. For example, this is the one pass receptor, and this one pass receptor is called insulin receptor, right? And this glucose, for example, I have told you there that in interstitial fluids and in blood the glucose will present same amount, right? From here, this glucose, for example, this is the glucose, insulin, sorry, this is the insulin. It will attach to this insulin receptor. Why insulin cannot cross cell membrane? Why it cannot move inside the cell? Because molecular weight of the insulin is very high. Insulin is macromolecule, it is a very large molecule and cannot cross easily the cell membrane, so it will act on insulin receptor. And when it binds to insulin receptor, what will happen? All the processes, the phosphorylation processes, which we have discussed in your first year receptors, right? It will undergoes phosphorylation. Phosphorylation means it will attach phosphate group to this vesicle, and by this vesicle will attach to this membrane. For example, it will attach here, and the GLUT4 receptor will open. The best way by which these vesicles are attached to the cell membrane is that, for example, here it will attach like this, and it will open their channels outside, right? And by this now it will take the glucose from outside into inside the cell. This is the mechanism behind GLUT4 receptor. Now, if insulin is not present, can these GLUT4 receptor work? No. So there are two types of diabetes mellitus. One is called diabetes mellitus type 1 and one is called diabetes mellitus type 2. So what is diabetes mellitus? What is diabetes mellitus? Increased sugar level or disturbed sugar level, right? So we have two diabetes mellitus. One is called diabetes mellitus type 1. Diabetes mellitus type 1, and second is diabetes mellitus type 2. What is the difference? What is the actual difference between these two? In diabetes mellitus, the pancreas is not able to produce enough insulin to take this glucose from the blood into the cell, and so the sugar level in the blood increases, right? For example, in this is the pancreas. This is the pancreas, and pancreas beta cells of pancreas not able to produce insulin. This is diabetes mellitus 1, and when this insulin is not present, this glucose cannot move from blood into the cell, so sugar level will be increases here, and this condition is called hyperglycemia or diabetes mellitus, right? What is diabetes mellitus 2? This is called insulin resistant diabetes mellitus. Insulin resistant diabetes mellitus, because the pancreas producing enough insulin, but there is a resistance to these receptors. For example, I am taking another cell here. This is the cell, right? And this is the insulin receptor, right? And insulin is producing by pancreas. enough insulins are present, but here are some resistant. For example, antibodies are produced against this receptor, and this receptor is blocked. This receptor is blocked. Insulin cannot bind into these insulin receptors, so GLUT4 receptor will not able to take the glucose from outside to inside. This condition is called diabetes mellitus 2. Basically, we are giving the insulin to the patient which have diabetes mellitus type 1, because in diabetes mellitus type 1, pancreas is not able to produce insulin, so we are giving insulin from outside insulin injections. But here insulin is present enough. We will prescribe that type of drugs to the patient, the medicines to the patient, which activate this insulin as well as this receptor, right? And when we will discuss glycolysis, you will see that this insulin is also responsible for increasing the glycolysis, because it is responsible for formation of a very special type of protein which is called hexokinases, or which is called glucokinases, right? Also it will form the 1,6-fructose bisphosphate kinases, or phosphofructokinase 1, right? And it is also responsible for making pyruvate kinase enzymes. All are protein, right? And enhance the glycolysis process. So this is the glucose transport system, and these are the two clinical points behind the GLUT4 receptors, right? Any question up to now?
The glucose regulatory pathway